Extracting DNA from strawberries is a relatively easy process that can be done in a few simple steps.
The first step in any experiment is, of course, to gather your materials. For this experiment, you're going to need a sandwich bag, a test tube, a pipette, a strawberry cut into quarters, a half-cup measuring cup, a teaspoon, a tablespoon, a thirds measuring cup, a cheesecloth, a funnel, and a small cup or glass beaker if available, a small bowl or another glass beaker if available, salt, rubbing alcohol, and dish detergent. The first step in this experiment is to get a half teaspoon of salt. So you're going to take your teaspoon and get about half. And you're going to put in a third cup of water. And then put in your salt. And you're also going to put in a tablespoon of Dawn dish detergent. Now we're going to mix that little concoction together as best you can. It is kind of hard to mix in the detergent because it's more dense than the other materials, but just do your best. It should end up looking something like that. And then our next step is to put your quartered strawberries into the plastic sandwich bag. And you're going to want to reseal it and just try and make sure you get all the air out. And you're going to want to mash the strawberries up as good as you can. You want to try and get it really mushed up because this is the process where you break the cell membrane and all of the cell organelles can come out, including the DNA that we're trying to extract. Now our next step is to add 3 tablespoons of the mixture you made first to your strawberry mixture. And you're going to want to mix that up. It's going to get a purple-ish sort of tint from the blue dish soap mixing with the red strawberries. Now our next step is going to be to put your cheesecloth in your funnel. And you're going to want to slowly pour this mixture . Now this can take anywhere from 2 to 5 minutes. You're going to want to let this liquid you see gathering at the bottom, you're going to want to let all of that drain out. By now, most of the liquid should have gone through the funnel and the cheesecloth into the bottom. It should look something like this. Now you can take all of the goop, as I like to call it, that is in the cheesecloth, and you can dispose of it. Now we are going to take this and use a pipette or any other instrument you have at home that you can use to sort of funnel things into a smaller container. And we're going to put it in the test tube. Now Now you're going to want to try and get the bubbles off the top because it's going to make it harder to see the DNA. There you go, we have that. And now we are going to take our rubbing alcohol. To put some rubbing alcohol in a half cup. You don't want to use the full half cup because as you see our test tube is a little smaller. And you're going to gently put that in. You can gently put it in as I have just demonstrated, or if you have a pipette, you can use that to put some in. You want approximately 1 inch layer of liquid, which that's about right. Now you want to put the cap on your test tube. Be sure not to shake it or try and mix it or anything because you want those 2 layers. And we're just going to let that sit for a little bit while the DNA forms. And now you've just extracted DNA from a strawberry. It's quite a simple process. And there it is. Thanks for watching.